Hello and welcome. Thank you everyone for joining us today for our second free CE lecture in our MyDog webinar series. This lecture is credited by the SCVMA, the Southern California Veterinary Medical Association, and to receive your certificate, please just follow the link posted in the chat. Here at MyDog, we use next-generation DNA sequencing and microbiome analysis to diagnose infectious diseases in all kinds of animal species and sample types. There's so much to learn about the microbial communities present in our pets, wildlife, and a lot of research is being conducted on the animal microbiome and its impact on health. In our webinar today, we will specifically focus on the microbiome of canine and feline species. It's an honor to welcome and introduce our today's speaker, Dr. Aline Rodriguez-Hoffman. Dr. Hoffman received her veterinary medicine and master's degree from La Universidad Federal de Santa Maria, Brazil. She pursued a combined veterinary pathology residency and PhD program at Texas A&M and was certified by the American College of Veterinary Pathologists in 2009 and received her PhD in 2011. She's currently an associate professor in the Department of Comparative, Diagnostic, and Population Medicine at the College of Veterinary Medicine at the University of Florida. Her research is mostly focused on studying the skin microbiome of companion animals, basic dermatology research, pathogenesis of infectious diseases, and the identification of fungal pathogens in histological specimens. She's a member of the American College of Veterinary Pathologists, the American Academy of Veterinary Dermatology, and the American Society of Microbiology. Dr. Hoffman has been incredibly busy and has published over 100 peer-reviewed scientific publications.
Papers in terms of skin microbiome was published by the Julia Sagres group from NIH back in 2011. I'll be honest with you, when I started in this field, it was super interesting because when you would type on NCBI, to look for papers on skin microbiome, about 12 papers would pop out. These days, if you type skin microbiome, you will get thousands of papers popping out. So the research has actually boomed over these many years. So I was able to follow as we progressed in this field. And then this hallmark paper was the first one to describe the composition of the microbiome, dividing the skin into different regions, into different niches. Some niches were more sebaceous, such as those in the glabella, the alarcres, the external auditory canal. Others were considered to be moist, such as, for example, the gluteal crease, the popliteal fossa, and the plantar heel. And then there were other regions described as more dry, such as the buttock, the volar forearm, and the hypothenar palm. So all of these regions had different types of composition. And then using next-generation sequencing, researchers were now able to describe all the organisms found within the skin surface of humans. They then moved into doing skin metagenome, identifying not only the bacteria, which the initial paper described, but actually all the organisms that they could find, all the DNA organisms they could find, including eukaryota. This includes primarily the fungi, as well as DNA viruses. And as you can see, when using metagenome, most of the composition is actually bacteria. A large number of these end up being DNA viruses, which are actually encompassed by phage. And then there is also the eukaryota that is usually a small proportion, except for the external auditory canal that had a slightly larger proportion than seen with viruses. An important thing to understand about the microbiome is that it's unique. Okay, so if we take this graph, this is called a PCOA plot. It describes beta diversity. So this is the comparison of all the samples. So each of the samples that were taken in this project, they are represented here by one dot. So if you think about that, these are all multiple dots. So these are different individuals that were collected over time, and then they were collected samples from different regions of their body. Now think that maybe we have, let's say, 100 dots or even more than that right here. So you can see that the gastrointestinal tract, people tend to be way more similar. So these dots, the closer they are together, the more clustering we see, the more similar the specimens are. The more further apart they are, the more likely the individuals are to be different. So Here, as you can see in the skin dots, which are represented here in green, you can see that the skin is quite variable. So today, if we are to collect samples from each individual attending this meeting, we are probably going to be able to go back and identify which sample belongs to each individual, because the skin microbiome is unique. There has been studies done nowadays with forensics, for example, trying to identify who used a keyboard, for instance, and that can be done again using this next generation sequencing to identify the composition of the microbiome. So there are different types of studies and uses that we've developed over the years based on these initial studies that describe the skin microbiome. So despite the fact that the bacteria or the fungi and the DNA viruses may change across individuals and across different regions, a lot of times the metabolites that are produced by the bacteria in these different regions, represented here in this graph above, they will be similar. So even though we have different compositions across our body in the different niches, we will end up having similar metabolic pathways that will be activated by these bacteria. So there is some compensation even when you change the composition of the microbiome. The skin microbiome is influenced by different factors. As you can imagine, it's exposed to the environment, so it's highly influenced by several different factors. One would be the physical factors of the individual, the biological factors, such as age, sex, genetics, species, breed, and maternal transfer. The environment, so animals kept in captivity versus those in the wild. Multiple studies have looked into individuals, humans living in rural versus urban regions, geography, individuals living in more dry climates versus humid areas. Pollution has been found to also be another factor influencing the skin microbiome, as well as the climate. And of course, of interest to all of us is the health status of individuals, right? Infection can result in significant change in the skin microbiome, as well as antibiotic usage. Multiple studies, especially focused on the gastrointestinal microbiome, have shown that the use of antibiotics is one of the main influencers in changing the composition of the microbiome within the gastrointestinal tract. Patients that have immune deficiencies, hyperimmunity, and alterations in the barrier integrity, such as those seen with individuals with atopic dermatitis and mutations in the filaggrin gene. Lifestyle, so diet, hygiene, cosmetics, and cohabitation has also been other factors influencing the microbiota. So I selected just a couple of papers to show how cohabitation, for example, can influence the human microbiome. In 2012, the Robert Knight's group published a study looking at cohabitation of people that lived with their dogs, and found that of the 60 families, there were several spousal units with and without children that lived with pets, and that the majority of the influence of the microbiome was within the skin, and very little was seen, or almost no significant changes in the oral cavity or the GI. So dog ownership described in this paper demonstrated that there was increased diversity of the microbiota in these adult individuals, not necessarily in the kids, and that the adults tend to share their skin microbiota with their own dog. So how did they find that? They found that some of the bacteria, taxa, that were found in people were then found in the dogs that were cohabiting with humans. Another study also looking at a wide variety of species. They took animals from the zoo, and then they also included in the study humans, dogs, cats, and then cats living with dogs, has found that a lot of times the cats living in households tended to cluster very closely to humans. And then The cats that had also a dog in the household, they tended to cluster more closely to the dogs. So the dogs, again, like to share the microbiota with whoever is living with them. Dogs have this behavior of going outdoors, shaking themselves in the grass, so they end up acquiring a lot of the bacteria and fungi that are within the environment, which then they bring into the interior of the household. In our house, these are pictures, old pictures from my oldest son. We are a firm believer of the microbiome, so we always made sure that our kids would be sharing, or that our dogs would be sharing their microbiota with our kids. So believing in that the shared microbiota would increase their diversity and then reduce their chances of developing allergic skin disease. And neither of my kids do have allergic skin disease, so we're lucky on that aspect. Now let's just touch a little bit on the canine skin microbiome. In 2014, after starting to follow the human literature, we were able, together with a group of researchers, at the time I was at Texas A&M University, so we developed the first project looking at the skin microbiome, trying to mimic what people have done looking at the human skin microbiome. And what we found was that dogs have very, very diverse skin. We looked at 12 different sites. We selected sites that we commonly see skin lesions. And then we found that most of the bacteria that were found in the canine skin were actually acquired from the environment. Interestingly, areas such as the dorsal-perianal region had some of the bacteria, which are here represented in maroon or red, that are the Bacteroidetes, and these are bacteria often found in the oral cavity or in the gastrointestinal tract. Same thing with the lip commissure and even the nostril and periocular region. Those are kind of regions of mucocutaneous regions. So they had a little bit different composition, also with increased numbers of Bacteroidetes. We found that the haired skin was more diverse than the regions that we consider to be of mucocutaneous junctions. So that's not surprising given the behavior of these dogs. And that some regions had a little bit more unique, especially as it relates to the mucocutaneous junctions. However, when we went to look into the different regions of the haired skin, there was a lot more shared microbiota across different regions of the body, so not following necessarily what was seen in the human study. We also looked at the fungi in the skin of dogs. This was done by a graduate student in the lab at the time, Dr. Courtney Misen-Smith. And we found again that the fungi in the environment, primarily Alternaria, Cladosporium, and Epicoccum, were found in the surface of these healthy skin dogs. We found that there was more clustering within the individual, but very little influence of the body sites that we selected for this study. So again, for the fungi, for the fungal communities, it tended to be more unique versus related to the region of the body we were selecting for the study. Other groups have looked at what is the impact of an early life skin microbiota on the development of canine atopic dermatitis. So this group from Switzerland, they evaluated the skin microbiota of West Highland White Terrier puppies and their parent dogs. So the parents had atopic dermatitis, so there was a high susceptibility for these puppies to also develop atopic dermatitis. They found that the microbiota differed between these different sampled sites, the age of these animals, of course, puppies versus their parents. The puppies also had differences in the season of sampling, the geographical location that the pups were coming from, and the hygiene status of the household, as well as whether these dogs had access to outdoor environment or were primarily kept indoor. However, they were unable, unfortunately, to find a core early life microbiota that could predict the development of atopic dermatitis. So they followed these dogs for quite a while until to evaluate if they were I think there was for at least a year or two, to see if they were already developing atopic dermatitis. And then of those that developed, there was no core microbiota. Certainly, this is something of an area of great interest to all of us, because imagine if you have an early core microbiota, you would be able to predict from a puppy's age whether that animal is more likely to be an atopic individual versus not. Certainly, this is an area that we need to continue to investigate and investigate further, because it could be definitely rewarding, to know and then try to use. If we are able to identify a core microbiota, we could then try to establish protocols trying to prevent the development of lesions that then go into chronicity. In terms of the feline skin microbiota, there are also several studies that have been done with, again, identification of a very variable and diverse skin, as was seen in dogs, although to a lower level than seen in dogs. And interestingly, what we found in one of these studies was that the composition of the actual hairy skin, remember that I mentioned this bacterial group called Bacteroidetes, these are often found in the oral cavity. Notice here, Bacteroidetes is almost 50% of the composition in the oral cavity. So in the skin of cats, we actually find a lot of these bacteroidetes throughout the hair, the skin, and we assume that that's because of the behavior of cats. Cats are constantly licking themselves. They are constantly licking their hair and their skin, and so they are spreading those bacteria within the oral cavity throughout their body and their hair and skin. This was a study, and you will see her picture in a few of these slides, that was done by another graduate student, Dr. Caitlin Older. We then looked at the cutaneous and oral microbiota and how it was influenced by breed and the environment. So the primary goal of this study, you can notice here that in this study, we selected some unusual breeds. We went into the literature trying to find breeds that are considered to be hypoallergenic. And so we selected some breeds that are anecdotally called hypoallergenic. And so we selected these breeds trying to see if perhaps not only the surface of their skin or the protein in their skin, which is often associated with being hyperallergenic versus hypoallergenic, but not only that, if perhaps the cutaneous microbiome could be one of the reasons that could influence the possibility of these potential breeds or anecdotal breeds being hypoallergenic. So we selected this group of cats, and at the same time, we used as our controls cats that were strays, and they had just been caught, that were primarily outdoor cats. And then we also had a group of cats that were exclusively indoor cats. So what we found is that for the breeds, we found difference in the number of taxa present, the communities that were present in this skin, as well as the relative taxa abundances. And we found significant difference in the relative abundances of Malassezia species. This was not surprising. This has been previously described for Devon rex and Cornish rex, that they tend to have higher relative abundances of Malassezia as compared to other breeds of cats. And this has been shown before with culture-based studies. So our study confirmed what had already been published for the culture-based studies. We found that Malassezia restricta and globosa were the most abundant. And then when we looked into the indoor versus outdoor cats, we didn't really see differences in the number of taxa present or necessarily the community structures. We were expecting to see the outdoor cats to be way more diverse than the indoor cats. That wasn't a finding that we observed, at least in this study that had a small sample size. I think we had about 10 to 20 cats total for the indoor versus outdoor. But we did see some difference in taxa abundances, where some of the outdoor cats tended to have way different types of composition, and the indoor cats tended to have some of the bacteria that are more commonly seen, for example, in human skin, such as Corynebacteria. We looked as well into the canine skin, the feline skin microbiota, and also found again that some of the fungi in the environment were also found predominantly in the skin of cats. So these are a lot of times found in the indoor environment. Now let's move on and talk a little bit about cutaneous dysbiosis. And when I say dysbiosis, what we mean is that alteration in the balance that we have in homeostasis of the composition of the microbes inhabiting the skin. So several factors again, we talked about some of these that they can influence, and the same factors that can influence can also alter the skin microbiome. One is lifestyle, whether people stay more inside or outside. The sports, for example, contact sports can influence the microbiome. Age, like as people age or as they go through adolescence in people, they tend to have different composition of the microbiota with high abundances of Cutibacterium acnes, previously known as Propionibacterium acnes, which then makes them more prone to develop acne at an adolescent age. Stress, communities, genetic predispositions, the diet that people are eating, hygiene habits, as well as, again, one of the main culprits is the use of drugs and antibiotics. All of that can result in alterations in the skin microbiota. And once the skin microbiota is altered, there will be imbalances in the communication between the microbes, as well as imbalances between microbes and the host. These altered interactions may lead to development of disease. In our caseload, we see primarily atopic dermatitis individuals. And then the development of disease with these continued altered interactions may end up giving these animals chronic disease. And so you go into that cycle that the altered interactions between the microbes and the host lead to continuous development of disease. And so you go into the cycle of patients having to be continuously treated because of these conditions that they have. The same group that I showed initially that described the first human skin microbiome study also looked at children with atopic dermatitis while they were flaring and then after they were treated. So these individuals, they were evaluated clinically, so at baseline as they were flaring and that post-flare. And what they found is that for the individuals here depicted in orange that were never treated, while they were flaring, the diversity of the microbiota, or which means the number of different bacterial species, that's all they looked in this study at the time, the number of different bacterial species reduced significantly while these patients were flaring. And then during the post-flare period, when they were getting better, the number of bacterial species now increased. In individuals that were intermittently treated, and treatment here included topicals, oral, so it was a wide variety of treatment that these patients had received, the diversity was actually maintained, as you can see here in these different lines in blue. So the changes in the microbiota, so let's look here at the controls. So here, again, human skin, highly diverse. The pink bars that we are seeing, these are Staphylococcus species. When they went and dug into what species were actually present in the skin of these individuals, these were primarily Staphylococcus aureus, the Staphylococcus epidermidis, sorry, for the control individuals, which are here in dark blue. When they looked at the individuals that had atopic dermatitis, what they found is that they had very high abundances of Staphylococcus, just like our controls. However, the children with atopic dermatitis actually were primarily composed of Staphylococcus aureus. In the individuals that were not treated and that were while they were flaring, you can see here that the abundances of Staphylococcus aureus increased significantly. and then here just Staphylococcus. In those that were treated intermittently, those amounts, those abundances of aureus were kept slightly down, the same as baseline. And then during the post-flare period, these individuals returned to what was seen at baseline. And what they showed in this study is that as microbial diversity went down, the proportions of Staphylococcus went up, and the proportions actually went up a little bit before disease severity was seen. So when these children had again, were they flaring up? After flaring, when the flare resolved, then the severity of lesions reduced, and then microbial diversity went up, and so the proportions of Staphylococcus went down. They also showed in an animal model that has it's called this ADAM17 mutation. This mouse model has alterations in the skin barrier, and so the bacteria on the surface can penetrate much deeper than what's seen with the wild type mice. So, and they showed in these individuals that they have much higher abundances, like in this case with Staphylococcus colony forming units using based on culture, and then with high abundances of Staphylococcus aureus on the skin of these mice, as well as Corynebacterium. So both were concomitantly increased in these mice with this mutation. And so when you have a normal skin that has high microbial diversity, you tend to have, again, large numbers of different types of bacteria. However, in these individuals that have an impaired barrier, when they have this impaired barrier, in this case, again, they used a mouse model, it resulted in activation of EGFR signaling leading to dysbiosis. So in this paper, what they showed is that the dysbiosis, actually the bacteria in the skin, was driving the inflammation in the individuals with atopic dermatitis. Certainly, these patients already had an alteration in the skin barrier, but it was the bacteria that was leading to the development of dysbiosis and inflammation in the skin. Now, in terms of dogs and dysbiosis, we know that our patients have a lot of allergies, especially atopic dermatitis. These individuals with allergic dermatitis, or more precisely atopic dermatitis, are very prone to secondary infections. Most of these secondary infections are due to pyodermas caused by Staphylococcus pseudointermedius, and then dermatitis often caused by Malassezia species, with these individuals have been prone to also developing external otitis. Here just some pictures from this paper showing different types of lesions. We see areas of alopecia and erythema around the ears, around the eyes, around the perioral region, sometimes even with crusting. A lot of lichenification and hyperpigmentation in the skin, and a lot of crusting on the skin surface as well. So we did some studies looking at these. Other groups have also looked, and then one of the papers that I wanted to highlight was published by folks from University of Pennsylvania, from Penn State VetMed, that Dr. Bradley published. Was that these patients had canine atopic dermatitis. They had very low diversity of on the surface of their skin, and they had high transepidermal water loss while they were developing flares of atopic dermatitis. And then with antibiotic treatment, these patients had remission of lesions. The diversity on the surface of the skin was restored, and so was transepidermal water loss, with significant lower proportions of Staphylococcus pseudointermedius. We also looked at superficial pyoderma in other groups of atopic dogs, and trying to see if the different types of lesions would be composed by different types of bacteria. So, for example, we looked at bacterial folliculitis and epidermal collarettes, and then we found that the epidermal collarettes had high abundances of Staphylococcus. This wasn't statistically significant. It was just a trend that we could see with high abundances of staph in the epidermal collarettes. And then with the bacterial folliculitis have lower proportions, but it could just be that we were swabbing the surface of the skin and not really going very, very deep into the follicle of these dogs that had bacteria. And then we tried to identify if there were differences in the species actually within these areas of bacterial folliculitis versus collarettes. And in most of these, it was really Staphylococcus pseudointermedius that was affecting the skin of these patients. When we looked at the fungal microbiota in allergic versus healthy dogs, and I say here allergic, not just atopic, because some of these dogs could have had other types of allergic skin disease, not necessarily atopic, so it encompasses all the types of allergic skin disease that we see and not only atopic dermatitis. So we found that the allergic dogs had lower diversity, they had different composition between the interdigital space and the ear canal with clustering among the dogs that were healthy, separately from the dogs that were allergic. So here you can see again in these PCOA plots. When we looked at malassezia, we found that approximately of all the samples we collected, only 6% were malassezia, but no difference were seen in the malassezia between the healthy dogs and the allergic dogs, at least in the study for the selected animals we sampled in this particular study. So then we digged a little bit deeper. We took all the sequences from malassezia and trying to compare whether the relative abundances of those sequences would change when we looked at the species level. And indeed, it was significantly different, where healthy dogs had very low proportions of Malassezia pachydermatis. They have been primarily composed of Malassezia globosa and restricta. And then when we looked at the allergic dogs, those tended to have much higher proportion of Malassezia pachydermatis as compared to the healthy dogs. So here just plotting all the samples that we collected from all of these animals. The My Dog Animal Diagnostics has also done a very interesting study, which was published in 2020 in Veterinary Microbiology. This has been the largest study that has been done so far, focused on the skin and ear microbiome. And in this study, they collected 600 samples to evaluate the skin microbiome of animals. This was the study that they actually used initially as their baseline to start developing these diagnostics of next generation sequencing for diagnosis of skin dysbiosis in dogs specifically. So in this study, they looked at both bacterial 16S ribosomal RNA sequencing as well as fungal YTS sequencing with more than 300 skin swabs from general swabs, about half from healthy individuals and half from lesional skin, and then approximately a little over 250 ear swab samples, being almost half of each, again, healthy versus lesional skin. And then what they found in their study was that the healthy individuals, they had very variable numbers of both bacteria and fungi. You can see here the samples from the ear, here the samples from the skin. And even though I'm not depicting what each color means, you just have to understand that these represents different bacterial taxa. So you can see a lot of colors here. When they look at the clinically affected animals, now you can see that these different samples pretty much are taken by a single taxa. Like all the colors that we saw here are no longer present. All we are seeing is a very small proportion of diverse bacteria, and most of this is now taken by a single taxa, which were characterized by Staphylococcus, primarily Staphylococcus pseudointermedius. And then in the ear, the same thing was seen, which was mostly Malassezia. The same thing was seen, even though it wasn't to the same level as the ear, but in the skin, they also saw significant loss of diversity for both bacteria and fungi. And then they looked at the diversity within these samples. And they also looked at the beta diversity. Again, this is another view for a PCOA plot showing that each dot represents a single sample. And then there was some clustering here among the clinically affected samples. And then here the healthy samples are clustering more with more widespread and variable composition for the skin samples. When they looked at the absolute numbers, because those numbers are usually based on relative abundance, when they look at the absolute numbers doing PCR targeting bacteria, which is here represented by the 16S gene, or fungi here represented by the ITS gene, they confirmed that not only the relative abundances were altered, but also the absolute abundances with clinically affected individuals have much higher absolute numbers of bacteria as well as fungi. Now, in terms of cats and skin dysbiosis, we've been able to also demonstrate similar trends for cats, where the allergic cats tend to have higher proportions of Staphylococcus. So although cats are not as prone to pyodermas as we see in dogs, cats also have dysbiosis of their skin when they are affected by allergic dermatitis again, or atopic-like skin dermatitis. And then when looking at the different types of Staphylococcus within feline skin, we found that a lot of this Staphylococcus, very little is actually composed of Staphylococcus pseudointermedius. This is mostly in the healthy individuals. And then the allergic individuals had high proportions of Staphylococcus capitis, epidermis, Staphylococcus felis, hominis, and as well as other less abundant species. Now we'll move on to another section in our talk, focusing on the use of next-generation sequencing for diagnosis of skin dysbiosis. When I gave my first microbiome talk, that was back in 2014. I remember I included in my talk what was the long-term goals of skin microbiome research. I had my short-term goals and my long-term goals, and this specifically was one of my long-term goals for skin microbiome research, was to be able to have assays that can diagnose skin dysbiosis. I'm very glad that we have reached this point now that we can do assays such as the MyDog that can provide a whole overview of the microbes that are within the skin. So at least one of my long-term goals back from 10 years ago have been accomplished, and we now have affordable assays that can be used to diagnose skin dysbiosis. So how does these methods really work? So I decided to include a little bit here on how these assays are done, and how we can identify those bacteria that a lot of times, or bacteria or fungi, may not necessarily be identified using culture. So these assays, they are targeted. So they may be done based on next generation sequencing with targeted regions. So a lot of times we do bacterial targeted regions, primarily focused on this gene, the 16S gene. So there are different regions that we target within this gene, which are the V1 and V3 region, as well as the V4 region. We've used these different regions in our studies before. And then for the fungi, we have a lot of times we target the 18S ribosomal RNA gene, or the ITS, that's an internal transcriber spacer region. Again, we use primers that target these different regions. These primers amplify these variable, hypervariable regions that you can see here within these bars. And then we put that into a phylogenetic tree, trying to identify what those organisms are, and that's how we can identify multiple organisms using these assays. Another way to do these types of assays is actually doing whole genome sequencing. In the case of whole genome sequencing versus targeted sequencing, we identify all the DNA within the sample. That includes a lot of times the host DNA, the microbial DNA, eukaryotic, the fungi, as well as the DNA viruses. And in talking to Janina, she has mentioned to me that my dog will actually be moving to whole genome sequencing in the upcoming future. So you might be seeing, for those that send samples, that in the future they might change to a more a whole approach of all the microbes, including as well as the host, but all the microbes within the sample, being able to identify protozoa, and parasites, other parasites. So a little bit about next generation sequencing. So we use these sequencers. There is I don't get any. I'm not supported by Illumina. I don't get any money to put their picture here, but it's one of the sequencers that's commonly used. There are several others that are used nowadays, and then the sequencers, to me, they work just like a diver. So you want to get a lot of sequences. The more sequences you get, the better comprehension you will get from the sample you are evaluating. So in a diver, if a diver is diving on the surface of the water or on the surface of the sea, you will see the very abundant fish, right, that are within the sea. However, as the diver goes deep into the sea, then it's more likely to see the rare fish that you're not going to see on the surface. So it's the same for sequencing. We try to sequence as much as possible. We try to get as many sequences as possible to be able to identify the rare species that could potentially be the triggers or give predisposition to certain individuals to develop disease. So the sequencing workflow, you in your clinic go and collect samples from dogs that have skin lesions. You then send that sample to the lab. They extract the DNA from that sample, and then they use currently this targeted approach targeting both the bacterial and fungal ribosomal RNA regions. So they use the primers targeting conserved regions, and then followed by sequencing that will be sequencing the variable regions. They then amplify all of that, again trying to go very deep so we can find the rare species. And then from that, we do in our lab, and just like they do at my dog, a lot of data processing, quality control. You have to reduce a lot of the junk that comes with it. And then you align all the sequences that are present. So if you have a dog that, let's say, has 90% of Staphylococcus species, that's all based on sequencing. So you would be getting sequences that would all be similar, and so you align all of them. We use like databases to identify these different sequence variants, and then followed by taxonomic classification, evaluation of community and phylogenetic analysis. This is primarily used for research, as well as relative abundance and composition, looking at diversity of the skin microbiome. So one study published by my dog group was a study that showed that next generation sequencing had advantage over traditional culture. In my opinion, one assay does not exclude the other. The more assays we can run and can offer to our clients, the more likely we are to get deeper in identifying what's the cause of certain skin lesions and skin infections we are seeing in our patients. So in their study, they collected samples from 78 exotic animal species. They did both conventional culture testing as well as NGS analysis. So these were all from exotic animal species. So they took two separate swabs that were used for this different purpose. So their study included several bird species, mammalian, one fish, and then quite a lot of reptiles from different types of samples. And then what they found is that for the bacteria, both next generation sequencing approach as well as fungal as well as bacterial culture approach were good in identifying and reporting the organisms present in those samples. However, for the fungi, there was not a really good correlation. There was actually a very low correlation where NGS was able to identify a pretty significant abundance of fungi within the skin, and then and fungal culture was not as much, like identifying what could not be identified, for example, with next generation sequencing. So in terms of fungal identification, next gen sequencing was way superior. In addition, about 14% of the samples organisms could have been identified with next gen versus as opposed to culture alone. Here are some of the organisms that they have identified. And then this study was previously shown in the previous webinar given by Dr. Rosenkrantz, and then where he showed this patient that had Burkholderia gladioli deep pyoderma. So to give you an overview of how they worked up the case, so this was a 7-year-old golden retriever, had a deep pyoderma on the paw. You can see here the lesions taken by presented in the paper. I believe it was Dr. Rosenkrantz that took these pictures, and then the lesions started following treatment with cyclosporin and prednisone because this dog had immunomediated hemolytic anemia. So they did ancillary tests. So they did cytology, found bacteria within the cytology, lots of neutrophils as well as macrophages. And then they did skin biopsies, identifying that there was a fairly severe paniculitis in this specimen, very severe paniculitis composed of neutrophils with scattered macrophages. Histologically, they could see bacteria as well that was confirmed with gram-positive coccobacilli. Then they did aerobic culture. They found that the organisms were Pseudomonas-like bacterium that was resistant to antimicrobial classes, but susceptible to fluoroquinolone. They sent the sample to my dog, did NGS, targeting the V1 and V3 region, as well as the ITS2 region, and they detected a very high abundance of Burkholderia gladioli. They further confirmed this with shotgun metagenomic sequencing, and at least in their assay, they didn't find antimicrobial resistant genes. Burkholderia specific PCR was also run. Samples were sent to CDC and they were positive. And then after the PCR being positive, they took that PCR product, sent for sequence analysis, and found that it was 100% homologous to Burkholderia gladioli, confirming this suspicion in this case. The dog was treated with marbofloxacin and improved the lesions shortly after. I don't remember exactly how long it took, but the dog recovered at least. Now, the final part of our talk, we'll talk just a little bit about how treatment for cutaneous lesions can affect the skin microbiome. We've looked at the use of diluted bleach baths. As you know, this is one of the recommended treatments for dogs with atopic dermatitis. So together with Dr. Panovic from UGA, we wanted to evaluate if there would be an impact in the microbiome of these dogs. These are healthy dogs. They do not have atopic dermatitis. And fortunately, we didn't see changes in the microbiota when we looked at 4 different dogs. For this study, we didn't see any change in the microbiota, significant change with the use of bleach baths. So it should be something that could be used without many concerns, in terms of resulting in dysbiosis just by the use of bleach baths. Dr. Rexo and Bernstein, as well as together with the MyDog group, also looked at topical medications on the nasomaxillary skin fold microbiome of French bulldogs. So they had 6 dogs that were controlled, 6 that received this Calzyme. It's an inhibitor of biofilm formation without biocidal activity. And then 6 dogs that received 2% chlorhexidine diacetate solution. The nasal folds were scored, and then all of these dogs were systemically healthy. So they found was that there were changes in the diversity scores. So both the enzyme and chlorhexidine improved the diversity of these animals. The enzyme improved slightly a bit more. And then the relative abundance of relevant pathogens actually decreased again with the enzyme and the chlorhexidine baths. So demonstrating again that both enzyme and the chlorhexidine, they do improve this biosis, reducing the abundances of pathogenic bacteria in the skin. We all have also been interested in canine body odor. I've always, I've attended skin microbiome meetings before, and at one time in one of these meetings, I was able to meet a researcher, Chris Kellyward from University of Ghent, that does a lot of armpit microbiome. I think we all are familiar with this. Some of us will have a little bit more of an odor, in the axillary region, and it is partially in great part related to the microbiome that resides within our armpits. So this group has focused on identifying the clusters and the types of bacteria that we have in the armpit microbiome, with high abundances being found for Staphylococcus and Corynebacterium. They've also done studies, interesting studies. When you go to the gym and you have this polyester fabric and then you come back stinky. So that is because polyester is actually the type of fabric that selects for this bacteria to proliferate. It does allow, it's permissive for this stinky bacteria to proliferate. The other thing that they found is that when we wash clothes with normal wash cycles at about 30 degrees Celsius and mild detergents, those may not be sufficient to remove all the bacteria that have been adhered to the clothes. So you may have noticed sometimes you wash your clothes, like the gym clothes, and then you finish drying it, and then when you take it out, and it's still stinky. It is because sometimes that the bacteria actually sticks to the clothes. So these studies have sparked our interest to also look at odor in dogs. And a few years back, we looked at dogs that had high odor. These were dogs that were these were hunting dogs, and hunting dogs, pseudo hunting dogs. They were used for sniffing and finding things. They were all of the same breed. And then on day 0, we all went there, we sniffed all these dogs, then we applied this Essential 6 Dermoscent, and I did receive funding from Dermoscent to do this study. It wasn't Dermoscent's idea to do this study, it was our idea to do this study, and we asked them if they could provide some funding for us. So, but just to declare here a conflict of interest is that they did provide the funding for this study. And then after 4 days, we went back after application of this product. We went back to the place where these dogs were housed, and we collected samples from this bloodhound and did the same type of microbiome assays. So what we found was that the dogs with the strong odor had a lower diversity of their microbiome. And then we identified 2 bacteria responsible for the odor, which were Microbacter and Pseudomonas. Those were significantly different between the dogs with strong odor versus no odor. Here, just whenever we extracted just those sequences, you can see here the proportions and significant differences, and which was mostly related to Microbacter, whereas Pseudomonas was just low abundances. When we apply those essential oils, here baseline again in red, 4 weeks application in yellow, we found that there was increased abundances for Pseudomonas with the control. The essential oils reduced them, but the placebo also reduced. So it's unlikely that had any effect on the Pseudomonas. However, for the control for Microbacter, there was some decrease in the essential oils group after 4 weeks, whereas the placebo there was actually an increase. The data wasn't significant. It was more of a trend that we found. So we weren't able to necessarily alter to the level of significance, but it's possible that the essential oils, either they reduced a little bit the Microbacter and that was what impacted the odor, or maybe just I would say perhaps hid the odor in the skin of those dogs. An interesting thing, one of the dogs that received this treatment was actually a department head for a small animal clinic. And then he was very thrilled with the product actually for his dog. And he said that was one of his complaints. The dog belonged to his wife and the dog was very stinky. And then after that, they were very thrilled, with the results that were obtained. And the dog was actually smelling really good in the household. So now, briefly, just microbiome as therapeutic target, just to conclude. Multiple groups, especially in human medicine, are looking into selecting commensal bacteria that can protect against Staphylococcus aureus. One is the group from Dr. Richard Gallo, which we have the pleasure of collaborating on another project. So they take this coagulase negative staph that are selected from skin swabs from normal individuals. They condition them to media, and then they put Staphylococcus aureus in the culture, and so they select the ones that can inhibit growth of Staphylococcus aureus. So one of these has been Staphylococcus hominis, and it's been currently used undergoing clinical trials as a bacterial therapy for Staphylococcus aureus in people, with antibiotics being the antimicrobial peptides present in these. These are mice. Presented with lesions. Other groups have also looked at Roseomonas mucosa, finding similar beneficial results and with inhibition of Staphylococcus. You can see here application of Roseomonas has reduced significantly the abundance, the proportions of Staphylococcus aureus in the skin. This has undergone clinical trial. There was a company that ended up getting the patent. They did a few more studies and the results were not as significant as was initially found. And I think right now the company has this on hold. They are not yet selling it for like treatment of atopic dermatitis. They might be looking at these more as a cosmetic product that could perhaps improve skin microbiota. I think it's just like the legal issues with having a product undergo approval for atopic dermatitis is much more complicated. The hurdles are much more complicated, given that the data that they found with, I think was a phase 1 or phase 2, wasn't as significant as the initial study. We have been involved together with Dr. Richard Gallo on a study evaluating Staphylococcus felis, trying to do the same thing. So Dr. Gallo has identified, and his group have identified this strain of Staphylococcus felis that inhibits the Staphylococcus pseudointermedius, as well as several other Staphylococcus. I forgot to, ended up not including another graph that showed that it inhibited several bacteria, but it was a very potent antimicrobial peptide inhibiting Staphylococcus pseudointermedius growth. So here the control, here is the extract, and then here is Staphylococcus felis C4, the actual bacteria. This is just the protein extract. And then we evaluated this using skin explants. I think I failed to include the picture of the graduate student that worked on this. This was Dr. Mayani Fakin that did this study, and we looked at Staphylococcus felis and Staphylococcus pseudointermedius on these skin explants, and you can see here with histology, lots of bacteria on the skin surface. And when we mixed them both, what we found was that there was a significant inhibition of growth of Staphylococcus pseudointermedius. Here, I only included the picture of the protein extract. Similar results were found when we used the Staphylococcus felis bacteria. However, with the felis, you still see just a thin layer of cocci bacteria on the surface of the skin. So we would not be able to tell exactly who those bacteria were. But with the protein extract, notice the difference between here, only rare cocci observed, versus here where you have a huge layer of bacteria overlying the skin. So to conclude, it's already 4 o'clock. So in healthy skin, we expect to see a diverse microbiota, low abundances of Staphylococcus species. In atopic dermatitis or allergic dermatitis, as I showed for most of these studies, we have reduced diversity, high abundances of Staphylococcus pseudointermedius for animals, which often is believed based on mouse model studies is one of the triggers of inflammation, which is aggravated in patients that have impaired skin barrier. So nowadays, we have a much better understanding of how skin lesions result in dysbiosis and how different treatment modalities can impact the microbiome, restoring homeostasis. We've been able to identify some of the key players of the commensal microbiota, and perhaps one day we are not there yet. We might be able to select some of this commensal and be able to augment the commensal microbiota, reducing the pathogenic bacteria, such as being done in the human studies. They are way further than we are. And then these early skin microbiome studies have paved the ground for the development of diagnostic techniques based on NGS for identification of dysbiosis and identification of pathogenic bacteria, like is being done by the assay provided by my dog. Lots of people to acknowledge. A lot of these studies I show that I participated have been done at Texas A&M. I've been now at the University of Florida for 3 years, and I collaborate a lot with Dr. Domenico Santoro, our dermatologist here, and Dr. Abraham Pelisseri, a microbiologist here. So, and then lots of, we've received multiple research support of the year, so we are very thankful for all the support we've received. And now I would be very happy to take on any questions you may have.
Thank you so much, Dr. Hoffman. That was a fantastic presentation. Very, very interesting. I feel like I've learned a lot. We do have at least one question in the chat. If anyone has more questions, please feel free to post them now as well. We have Dr. Komal asking, if a change in microbiota does not result in disease, can it result in increase of the immune system somehow? And can these alterations have positive effects in any case?
Can you repeat the question?
Mm-hmm. If a change in microbiota does not result in disease, can it result in increase of the immune system somehow? And can these alterations have positive effects in any case?
If a change in the microbiota, it just depends what type of change, right? Are we talking a change where we are increasing the number of pathogenic bacteria? Yes, if we are making changes in the skin microbiota that may end up increasing the number of pathogenic bacteria, yes, that can definitely have a negative effect on the skin. Now, on the other hand, if we are talking about an increase in the number of commensal bacteria, non-pathogenic bacteria, it is believed that an increase in diversity is often associated with a more healthy disease status, especially as it relates to bacteria. So in my view, although I may not be able to prove or support this based on all these studies, but the belief is that yes, a change in the microbiota, increasing diversity, should be beneficial for the skin of individuals. Now, if an individual has alterations in the skin barrier and you increase their diversity, it may not be enough. Like, it may not be that just the microbiome can overcome all the problems that we have, that individuals have, like such as an animal that has atopic dermatitis. You may be able to control the dysbiosis, but the inflammation may still be there. Having said that, the study from Dr. Kobayashi showed that when they recovered dysbiosis in those mice improved their skin. So they had improvement of their skin lesions. I can stop sharing here. Don't know if I answered the question.
We have another one asking, is there interest in pursuing long-term alterations to the microbiome? Perhaps when an animal or human moves from one geographic location to another. I'm wondering about the implications for a meta-analysis that showed the oral microbiome is different for different countries.
Yes, we have done studies where we show differences, geographic differences. When we collected samples, I'm trying to remember correctly. I think there are studies in people that have looked at individuals that change, but it is believed that when you move, you could, depending on the climate, there could be change that could happen. These changes, I believe, are minor because in general, we tend to retain our microbiome. So a lot of the change that will happen in the microbiome, they tend to be transient. So there has been other studies that have been shown that the skin microbiome is very resilient. So with everything we do, we change some of the microbiota, the skin microbiota specifically. So like swimming, when you swim, you change your microbiota, but that actually lasts hours. When you do sports, contact sports, you change your microbiota. You acquire the microbiota from the individuals that are cohabiting with you. But again, that lasts hours. It's not long lasting. What is more likely to happen is that we actually retain our microbiota, and we haven't done that in dogs to know how much they actually retain. Dogs have a lot of variability. They have a lot of influence of the environment. They are not bathing themselves every day, so they're not really washing away, which is different than humans, right?
Mhm. The next question is, are you able to identify causative agents in autoimmune conditions, such as feline stomatitis?
We did a study trying to find that, and we didn't necessarily were able to find significant pathogens. We found some bacteria that were altered, but it wasn't like, oh, this is the culprit. You know, we did already a study on feline stomatitis, and it wasn't like outstanding the results that we obtained. You did a study too, right, Yanina?
Yes, we did. We looked at feline stomatitis versus the healthy controls, and we found several bacterial taxa, mostly of anaerobes, that were
Mhm.
significantly enriched in those patients. The fungi were present too, but they were by far not as impactful as they were in canine
Right.
stomatitis. So in canine stomatitis, we had a strong correlation between fungi and bacteria, which we did not see quite as much in the feline study.
Mhm.
I have a question too. So you're surely familiar with the dysbiosis index developed by Dr. Jan Sudolsky from Texas A&M
Yes.
for GI issues. Do you think it would be possible to develop a similar index for otitis or dermatitis?
I think it would be fantastic if we could do something like that. I worked with Dr. Sukadowski from Texas A&M. He's actually the person that trained me at the beginning of these studies. He was very supportive with a lot of our initial studies. We tried. I gave once the data. We had like a mathematician trying to that helped them, and I sent it to them. They couldn't because in the GI there were several bacteria that participated. In dogs, a lot of times we find it's mostly the Staphylococcus actually, but I think it would definitely be beneficial. And at the time when we looked into doing that, we only had a very few number of samples. So now, especially with your group having so many samples and having acquired so many samples over the years, I think it would definitely be beneficial to do something like that. Absolutely.
When you just Google search the term microbiome, a lot of different topics come up. Everything from aging, the gut-brain axis, fecal material transplants. I recently got an advertisement that will help against wrinkles as well. Do you see some of these challenges? Like, where do you see the field moving? What do you think some are some of the challenges that we as a community need to address? What is actually achievable versus what is wishful thinking to some degree?
I'll tell you something that may be funny. I think last year, South Park had an episode about the gut microbiome. I could not believe that South Park did an entire episode about the microbiome. So certainly this is a hot topic, right? As you said, it's everywhere. You see it in the literature, you see in magazines. So I think like many things, it's usually not one thing causing disease. It's always an imbalance. So, for example, I study primarily the skin. So, I cannot tell you that it's just the bacteria. The bacteria are there. The healthy dogs have Staphylococcus pseudointermedius in their skin. They carry Staphylococcus pseudointermedius. They carry all different types of Staphylococcus, and yet most of the healthy dogs usually don't develop pyoderma. So to me, it's a combination of things. So I do believe that there is a lot of prospects in using the microbiome, in augmenting the microbiome to help as another treatment to improve the lives of individuals that have especially atopic dermatitis. So for instance, I can tell you in VetMed, we had, for example, a few years back, Zoetis launched Apoquel. Apoquel is a drug that's used to reduce pruritus, so dogs stop scratching. We thought that Apoquel would solve all of our problems because some researchers would show that it's actually the scratching that results in lesions. Apoquel came, it solved many of our problems, but not all. So there is not one single thing that we need to target. And I'm not on clinics, I'm mostly in a lab and I'm a pathologist here reading slides, but the folks that are on clinics know that it's usually a multi-targeted approach, right? We have to target all the different areas in the body that may have imbalances. And I think the microbiome is just another one because these dogs are prone to developing pyoderma. These cats are known to are also, although less, they also tend to develop overgrowth of bacteria, you know. And so if you are able to control some of that, you may be able to improve the quality of life of these patients, just like you improve the quality of life of patients when they stop scratching. So to me, again, it's a multi-targeted approach, and I think that the microbiome does have a lot of opportunities, again, for treatment. And as I mentioned before, what you were doing already, you know, in terms of diagnostics. I'm a diagnostician myself. And I think it's again, is this the answer to all of our diagnostic assays? No, because we may end up finding cases that we may find incorrectly identifying pathogens in patients that may not necessarily have that pathogen. So there is a chance for false positives. So it's the same thing for diagnostics. We should be able to combine, you know, the more assays that you do, the more likely you are to specifically identify what's the cause of lesions or what is one of the potential causes of infectious diseases in animals.
Mhm. We have another audience question. Is there a way to increase the diversity or competition for skin or ears, like skin or ear probiotics, for example?
Yes, there are a few products in the market. I don't want to mention them right now. Some of them folks here at UF are receive funding, but there are a few products in the market that are currently augmenting, again, increasing diversity. I have not worked myself with any of these products to say, okay, what is my experience, but there are a few products in the market, yes, that are at least trying to. Usually we haven't been very successful as with the gut in terms of having live bacteria probiotics. Probiotics should really be the live bacteria. We are not there yet as with the gut, because with the gut you give a load of bacteria and then it stays and you try to make it colonize. We haven't been done this as good as they've done, but yes, there are a few products, especially with bacterial products, not necessarily the bacteria, live bacteria, if I remember correctly. I could be mistaken, that could be something else, but for the bacterial products, for sure.
Mhm. Well, thank you very much, Aline. That was the last of our questions from the audience. I think it was a fantastic webinar. Thank you for joining us today. For the audience, if you would like to download your CE certificate, please use the link in the chat. We will also email everyone who has attended with the link or the attachment of the CE as well. So thank you all and have a great rest of your day.
Thank you everyone for attending, and thank you so much, Janine and my dog, for the invitation. I really appreciate and love sharing the research with all of you. Thank you.
It was wonderful. Thank you, Aline. Goodbye.
Goodbye.